A very pleasant day. We are from Group 13 from Biosensor Technology. We are explaining the colorimetric paper bioassay by horseradish peroxidase for the detection of catechol and resorcinol in aqueous sample. Catechol and resorcinol is a toxic and persistent water pollutant family compound. Catechol is classified as a possible carcinogen to human and toxic to aquatic animal, for instance fish, while resorcinol is endocrine disrupting chemical. In the previous study, researchers have utilizing multiple detection methods of this pollutant either by using conventional methods such as spectrometry and chromatography or an electrochemical biosensor for simultaneous detection or optical biosensor using lactase enzyme and chromogen MBTH. Among the drawbacks of this method of detection was sophisticated instrument was used in the conventional method, leading to only qualified staff able to operate the instrument in addition of the time consuming which take ample of time to complete the detection of the pollutant. Based on the amperometry and optical biosensor, these biosensors are expensive and complicated. So, what is a real problem that we intended to solve from previous study? The real problem are the usage of complex and time-consuming biosensor fabrication methods that proposed by researchers. Those methods has led to limited study on the specific response of sensor to the pollutant in real-time water sample. Hence, this problem can be solved by development of simple, cost-effective, portable, and disposable paper-based biosensor.
Next, moving on to significance of colorimetric paper assay. So this colorimetric paper bioassay uses horseradish peroxidase, a HRP, a type enzyme, for detecting catechol and resorcinol in an aqueous sample. It offers a simple and cost-effective approach for detecting this compound, as shown in the figure below, which can be an indicator of pollution or contamination in environmental sample. So this method involves immobilizing HRP on a Watsman filter paper through a physical absorption and optimizing cytosine concentration for enzyme entrapment. The calorimetric assay process include analyzing color intensity using ImageJ software. And lastly, the paper-based biosensor have the potential for various applications, including water quality, environmental monitoring, and point-of-care diagnosis.
This paper-based biosensor is very useful for real-time environmental monitoring of toxic compounds such as catechol and resorcinol. First and foremost, the advantage of using paper-based biosensor is because it is very simple to set up and use. The use of Whatman No. 1 paper as reaction platform has large pore volume that can incorporate the capillary action to allow easy flow of sample to enzymes for detection. The enzyme, horseradish peroxidase enzyme, is immobilized to the paper by entrapment in chitosan, which is biocompatible and does not interfere with the enzymatic reaction. This entrapment method also results in higher retention on paper, where it exhibits a microenvironment that maintains enzyme catalytic activity over a long time. The detection of catechol and resorcinol also easy, as users only need to observe for color changes from pink to red with the presence of the chemicals and corresponds to the concentration in the sample. Next, using paper-based biosensor is cost-effective both to producers and also consumers. These biosensors does not require sophisticated instruments such as HPLC to detect the chemicals. Since the biosensor is small and compact, only a small amount of enzyme is needed for each paper strip. As mentioned before, the entrapment method ensures the catalytic activity of the enzyme for a long time, further cutting the manufacturing costs. Moving on, this paper-based biosensor is also user-friendly because it is easy to use with clear and simple instructions. Therefore, users does not have to require special skills or run out the extensive analysis to detect the chemicals. The biosensor can be used by non-experts for instant detection. The provided color charts can indicate the concentration of the chemicals present in the sample. Lastly, the paper-based biosensor is also eco-friendly, where it does not use any electrical devices, and papers are chemically inert, biocompatible, and biodegradable. The biosensor can also be stored up to 3 weeks in 4 degrees Celsius. The working pH does not differ significantly from neutral, makes it more easy for sample preparation. All in all, the paper-based biosensor is very helpful for on-site environmental monitoring because it is a sample to answer biosensor. Not to mention, the limit of detection in aqueous sample for catechol is 50 mg per liter and resorcinol is 10 mg per liter.
Hello and hi, I'm Roy, and today I will present the working principle of the paper-based biosensor that utilizes horseradish peroxidase or HRP enzyme as the biosensing element and its ability to catalyze reaction involving phenolic compounds, catechol and resorcinol, which are the target molecules. Let's dive in. There are different methods to immobilize the HRP enzyme. This paper discusses the HRP enzyme entrapment in chitosan on paper, physical adsorption of HRP on paper, as well as covalent coupling using glutaraldehyde on both chitosan on modified and modified paper. The higher the color intensity, the more stable the immobilized enzyme. Therefore, the best immobilization method is HRP enzyme entrapment in chitosan on paper, where the chitosan entrap enzyme distributed over the porous paper surface that shows a microenvironment that maintains enzyme catalytic activity over a long time. Moving on to the transduction method. The paper was scanned using a desktop scanner every 15 minutes after the dispensation of the catechol. Then, the color intensity will be measured using ImageJ software. Now, let's talk about the detection limit. For resorcinol, the detection limit is 4.5 millimolar, while for catechol, the detection limit was 0.6 millimolar. These values enable the detection of even lower concentrations of phenolic compounds. The sensitivity of the biosensing technique is remarkable. With resorcinol, it can be observed a transition from faint pink to dark orange, allowing a visible limit of detection of 10 mg per liter. Similarly, for a catechol, a transition of faint pink to red was observed, enabling a visible limit of detection of 50 mg per liter. So that's it for my part. Thank you so much.
The next part is conclusion. In one sentence, the journals or the research shares a novel approach to applying colorimetric paper assay by adding horseradish peroxidase to detect cytosol and resorcinol, where the scientists immobilized this HRP in cytosine entrapment with glutaraldehyde cross-linking technique that requires 15 milligram per milliliter of enzyme for the contraction, and it needs to be maintained at the pH of 6 to 6.5. In conclusion, the paper assay constructs a cost-effective, portable, disposable, and stable biosensors, where only using a paper we can detect the cytosol and resorcinol found in various types of water samples without the need of any high working skills or high efforts. The last part is biosensor improvement, where colorimetric paper bioassay may produce a rapid and portable method to detect cytosol and resorcinol. This means there is no need to take the water sample into the labs, there is no need for an expensive instruments, there is no need for a very long and tedious work to detect the analytes. The second point is the incorporation of horseradish peroxidase into the paper matrix enhances the versatility and ease of use, which means everyone can use this paper bioassay, even though it's a non-skilled workers or lab technicians, because there is no need for a highly skilled techniques to perform this assay. Which means the last point is this study marks the importance of using colorimetric bioassay, uh, and this is opens up the possibilities for further advancements to produce a new product where everyone can use to detect many kinds of other analytes for further and future improvements. Thank you, Dr. Nur Rahim and Dr. Shafinas, for watching our presentations.